Today, we're exploring the key ideas from Cancer as a Metabolic Disease by Thomas N. Seyfried. This is an independent educational overview, enriched with our own integrative analysis and actionable exercises. Think of it as a guided reflection to spark your own insights, not a replacement for reading the book itself. The conventional understanding of cancer as a disease primarily driven by genetic mutations has for decades dictated research priorities and therapeutic strategies. Yet, despite immense resource allocation and technological advancements in genomic sequencing, the persistent rise in cancer incidence and the limited success against advanced metastatic disease challenge this dominant paradigm. A compelling alternative, rooted in the pioneering insights of early 20th century scientists, argues that cancer is fundamentally a metabolic disease. This perspective asserts that the myriad genetic aberrations observed in tumors are largely secondary consequences, rather than primary causes, of a profound and irreversible impairment in cellular energy metabolism, specifically mitochondrial respiration. From this vantage point, the chaotic proliferation and systemic spread of cancer cells arise from an adaptive shift towards less efficient, fermentation-based energy production, a primal survival mechanism activated when the cell's crucial respiratory machinery is compromised. This reinterpretation of cancer's origins offers a unified framework for understanding its diverse manifestations and, crucially, points towards novel, less toxic, and potentially more effective therapeutic and preventative strategies centered on metabolic vulnerabilities. The metabolic root of malignancy: unraveling cellular dysfunction. The prevailing view of cancer posits it as a genetic disorder, where accumulating DNA damage and gene mutations transform normal cells into malignant ones. This gene-centric model has spurred vast genome sequencing projects and personalized drug therapies aiming to target specific genetic abnormalities. However, the author contends that the thousands of genetic changes identified in tumors often represent mere epiphenomena, downstream effects rather than the instigating forces of the disease. Despite their technical sophistication, these genomic endeavors have yielded limited clinical benefit, raising questions about their monumental cost and resource allocation. The sheer volume of mutations, coupled with paradoxes like the frequent mutation of essential genome guardian genes, highlights inconsistencies within this genetic paradigm, suggesting a fundamental misapprehension of cancer's true initiation. In stark contrast, a consistent and universally observed characteristic of cancer cells is their altered energy metabolism, famously termed aerobic fermentation, or the Warburg effect. This phenomenon describes cancer cells' elevated glucose uptake and lactic acid production even in the ample presence of oxygen, a metabolic signature so robust it forms the basis for diagnostic PET scans. The core insight is that this aerobic glycolysis is not simply an inefficiency, but a vital compensatory mechanism for cell survival, necessitated by irreparable damage to the cell's primary energy-generating machinery. The author, building on these foundational observations, posits that profound, irreversible impairment of mitochondrial respiration is the prime cause of cancer. This respiratory insufficiency precedes and fundamentally underpins the genomic instability and other hallmarks traditionally ascribed to cancer. From this perspective, most observed genetic defects are not the initial drivers, but rather adaptations or consequences of compromised mitochondrial function, forcing cells to depend on less efficient, non-oxidative phosphorylation. Normal mammalian cells predominantly generate ATP through oxidative phosphorylation, OXPHOS, within their mitochondria, a highly efficient process accounting for the vast majority of cellular energy. A smaller fraction of ATP comes from substrate-level phosphorylation in the cytoplasm, glycolysis, and mitochondrial matrix. Cancer cells, however, exhibit a distinct bioenergetic profile, a profound reliance on fermentation reactions, particularly the Warburg effect. While total ATP production in malignant cells might appear similar to normal cells, its source differs drastically. They may even consume oxygen, engaging in what appears to be pseudo-respiration, but without the efficient ATP synthesis characteristic of healthy OXPHOS. This inefficiency stems from tangible defects. Tumor mitochondria often appear fewer, smaller, and morphologically abnormal, with distorted cristae, the crucial inner membrane folds where electron transport and ATP synthesis occur. These structural aberrations directly correlate with reduced OXPHOS capacity and increased malignancy. Proteomic analyses further confirm these deficiencies, revealing marked shortfalls in key OXPHOS enzymes alongside elevations in glycolytic enzymes. Additionally, the lipid composition of tumor mitochondria is often deranged, particularly concerning cardiolipin, a unique phospholipid essential for efficient OXPHOS. Such abnormalities in structure, protein, and lipid content are not random, but point to a fundamental impairment in respiratory energy production. The historical Warburg dispute centered on whether cancer cell respiration was truly damaged. Critics often pointed to observations of oxygen consumption in cancer cells as evidence against Warburg's theory. However, this argument frequently overlooked the crucial distinction between mere oxygen consumption and efficient coupled ATP synthesis. Tumor mitochondria can consume oxygen while simultaneously exhibiting increased proton leakage across their membranes, dissipating the electrochemical gradient as heat rather than converting it into ATP. This uncoupling results in inefficient energy production, forcing a greater reliance on glycolysis. Furthermore, many studies attempting to refute Warburg failed to adequately differentiate between ATP derived from OXPHOS and that generated by mitochondrial fermentation, particularly from amino acids like glutamine. Glutamine plays a critical role in cancer cell energetics, serving as a major fuel source for ATP production through substrate-level phosphorylation within the TCA cycle, especially when OXPHOS is compromised. This glutamine fermentation can mimic OXPHOS activity, leading to misinterpretations in experimental findings. Methodological flaws, such as using inappropriate cell comparisons or growing cells in high glucose media that can induce glycolytic shifts even in normal cells, have perpetuated confusion. A rigorous examination under identical, respiration-favoring conditions consistently reveals impaired respiratory function in tumor cells. Crucially, the genomic instability, often seen as cancer's defining feature, including oncogene activation, tumor suppressor inactivation, and widespread mutations, is reinterpreted as a consequence of this fundamental respiratory defect. When mitochondrial function is compromised, cells activate a retrograde RTG response, a mitochondria-to-nucleus signaling system that epigenetically shifts cellular metabolism towards fermentation to ensure survival. This response upregulates oncogenes like MYC and RAS, which facilitate fermentation, and simultaneously disrupts DNA repair mechanisms, leading to genomic instability. Chronic inflammation, a known carcinogen, damages mitochondria and drives this metabolic shift, often stabilizing HIF-1 even in oxygen-rich environments, pseudo-hypoxia, further entrenching the cell's reliance on glycolysis. Healthy mitochondrial function is vital for DNA repair and mitotic fidelity, making respiratory insufficiency the real mutator phenotype. Even aneuploidy, abnormal chromosome numbers, is linked to calcium dysregulation stemming from mitochondrial damage. The ultimate proof of mitochondrial primacy comes from nuclear cytoplasm transfer studies, where tumor cell nuclei transplanted into normal cytoplasm with healthy mitochondria fail to induce malignancy. Conversely, normal nuclei placed into tumor cytoplasm often become tumorigenic. This striking evidence underscores the cytoplasmic, specifically mitochondrial, control over the malignant state, positioning the mitochondrion as the ultimate tumor suppressor. The far-reaching implications of mitochondrial dysfunction extend beyond the primary tumor, fundamentally reshaping our understanding of cancer's systemic impact, particularly metastasis, the process responsible for the vast majority of cancer-related deaths. Many traditional cancer models, despite their technical sophistication, fall short in replicating the complex realities of metastasis. Injecting tumor cells directly into the bloodstream, for example, bypasses crucial physiological barriers, much like baiting a mousetrap with a picture of cheese rather than actual food. Xenograft models, using human tumor cells in immunocompromised mice, introduce further distortions. Human cells acquire mouse biochemical traits and operate within a vastly different metabolic environment, creating human mouse centaurs whose responses may not faithfully reflect human disease. Even genetic models often require multiple simultaneous defects to induce limited tumor growth, rarely mimicking the widespread invasion seen in human cancer. Cell culture models, while useful for specific molecular mechanisms, also struggle to predict invasive behavior in a natural biological context, often failing to account for the dynamic microenvironment. More accurate natural models, like specific mouse strains that spontaneously develop metastatic tumors, offer a clearer path to understanding disease progression by focusing on biological behavior rather than simply origin or histological classification. The various hallmarks of cancer, characteristics once thought to define the disease, are reinterpreted through a metabolic lens as direct consequences of insufficient mitochondrial respiration. Uncontrolled cell proliferation, for instance, is not a random event, but a default state for cells with impaired respiration, which activate the retrograde RTG response and upregulate oncogenes like MYC and RAS to sustain fermentation. Telomerase activity, associated with cellular immortality in cancer, also appears to be a consequence of this metabolic shift, potentially relocating from dysfunctional mitochondria to the nucleus. The evasion of programmed cell death, apoptosis, another hallmark, is a physiological adaptation. Only cells capable of shifting to fermentation can bypass apoptosis, as the pathways sustaining fermentation, e.g. mTOR, MYC, RAS, HIF1-alpha, are inherently anti-apoptotic. Furthermore, the sustained angiogenesis, or new blood vessel formation, critical for tumor growth, directly supports the metabolic demands of fermentation. Tumors require this enhanced vascularity to deliver glucose and glutamine and to remove toxic byproducts like lactic acid. HIF1-alpha, central to glycolysis in low oxygen conditions, also powerfully stimulates angiogenesis, illustrating how this metabolic shift drives multiple facets of the disease. Perhaps the most radical reinterpretation concerns metastasis. The widely accepted epithelial to mesenchymal transition, EMT theory, posits that epithelial cells acquire genetic mutations, transforming into mobile mesenchymal-like cells. However, the dramatic reversibility of this process, MET, required for secondary tumor formation strains credibility when viewed solely through a gene-driven framework. Instead, the metabolic theory proposes that metastatic cancer cells originate from myeloid cells, such as macrophages, or from fusion hybrids formed between neoplastic cells and myeloid cells. Macrophages are intrinsically mesenchymal, equipped with inherent capabilities for migration, shape-shifting, phagocytosis, and cytokine secretion, all traits characteristic of metastatic cells. This hypothesis elegantly bypasses the need for complex, stepwise genetic alterations to explain invasive behavior. Compelling evidence supports this myeloid origin. Many human metastatic cancers express macrophage-specific antigens and exhibit phagocytic activity. Animal models, such as specific glioblastoma cells, display striking similarities in morphology, phagocytic activity, and metastatic potential to macrophages. The cell fusion hypothesis suggests that cancer cells, especially in inflamed microenvironments, can fuse with host macrophages. These hybrid cells would then inherit the macrophage's migratory and invasive prowess alongside the cancer cell's proliferative potential, potentially evading immune surveillance. This phenomenon is supported by observations in human cancers, particularly in bone marrow transplant recipients, where metastatic cells have been found containing DNA from both donor myeloid cells and recipient tumor cells. The seed and soil hypothesis of metastasis, where cancer cells preferentially colonize specific distant organs, also finds a coherent explanation. Macrophages are naturally programmed to enter and engraft specific tissues or sites of inflammation. If metastatic cells are derived from macrophages or their hybrids, their inherent homing patterns would dictate metastatic sites. Even the existence of transmissible metastatic cancers in animals further supports a macrophage epithelial cell fusion origin, with mitochondrial dysfunction driving their malignant phenotype. The remarkable genetic heterogeneity seen in metastases, far from being the primary driver, is thus understood as a downstream consequence of fundamental mitochondrial dysfunction and the varying genetic makeup of different fusion hybrids. This metabolic, macrophage-driven view of metastasis offers a powerful framework for developing targeted therapies that exploit the distinct metabolic and behavioral vulnerabilities of these highly aggressive cells. Rewriting the treatment paradigm. Targeting metabolic vulnerabilities. The entrenched reliance on conventional cancer treatments, surgery, chemotherapy, and radiation has produced limited success against advanced metastatic cancers, with annual death rates remaining stubbornly high. These aggressive interventions, while sometimes effective for localized or benign tumors, frequently inflict severe systemic disorder, accelerate aging, and compromise a patient's overall health, leading to ambiguity about whether death results from the disease or the toxicity of its treatment. Newer immunotherapies, despite their exorbitant cost, often offer only marginal extensions of life, accompanied by significant adverse effects. A critical assessment reveals that these gene-centric approaches, largely ignoring tumor energy metabolism, are unlikely to provide long-term solutions. The standard of care for aggressive cancers like glioblastoma multiforme, GBM, exemplifies this critical flaw. Despite aggressive surgical resection, radiation, and chemotherapy, often compounded by high doses of immunosuppressive corticosteroids, median survival remains abysmal. This approach inadvertently fosters an environment conducive to tumor progression. Radiation induces inflammation and elevates glutamate, fueling glycolysis. Corticosteroids drastically raise blood glucose, a primary fuel for fermentation-dependent tumor growth. And anti-angiogenic drugs can paradoxically worsen necrosis and increase invasiveness by selecting for cells that thrive in hypoxic environments. This creates a perfect storm, where standard interventions, through their impact on cellular metabolism, actively contribute to the disease's unmanageable progression. The persistent reliance on toxic, often ineffective therapies stems from a fundamental misunderstanding of cancer's metabolic origins. If cancer is indeed a metabolic disorder, characterized by impaired cellular respiration and an over-reliance on fermentation, then effective management must logically target these metabolic vulnerabilities. This paradigm suggests that therapies focusing on tumor cell energy metabolism could be universally applicable, independent of a cancer's histological origin, given the shared underlying defect of damaged respiration. A key strategy involves manipulating the availability of essential metabolic fuels like glucose and glutamine alongside synergistic interventions to exploit these inherent weaknesses. Dietary energy reduction, encompassing calorie restriction and specific dietary regimens, stands as a foundational metabolic therapy.
Extensive research consistently demonstrates that DER significantly curtails the growth and progression of diverse tumor types by primarily lowering circulating glucose, a critical fuel for many cancers. DER, distinct from starvation, involves a controlled reduction in overall nutrient intake without inducing malnutrition, thereby enhancing general health, preventing tumor formation, and diminishing inflammation. Its ability to reduce tumor growth without the severe adverse effects of conventional therapies makes it particularly appealing, even mitigating the toxicity of certain chemotherapies.
A crucial insight is that the anti-cancer action of DER primarily stems from the reduction of overall calories, rather than the restriction of any specific dietary component. Lowering total food intake, whether through a high carbohydrate or a high-fat ketogenic diet, effectively reduces aggressive tumor growth. This leads to reduced circulating glucose and elevated ketone bodies, such as beta-hydroxybutyrate. Ketone bodies serve as an alternative, highly efficient fuel for normal tissues when glucose is scarce. The efficacy of ketogenic diets, KD, in cancer management is maximized when consumed in restricted amounts, KDR, which actively lowers blood glucose and promotes the efficient utilization of ketones for metabolic energy rather than their excretion. Unrestricted consumption of a KD, if it fails to sufficiently lower glucose, may not yield therapeutic benefits. Normal cells possess the metabolic flexibility to adapt to reduced glucose by switching to ketones for energy, a naturally conserved evolutionary response to food scarcity. In stark contrast, tumor cells, with their compromised mitochondrial respiration, generally lack the enzymatic machinery or functional mitochondria to effectively utilize ketones. This metabolic inflexibility renders them highly vulnerable to glucose deprivation, while normal cells are protected and even energized. This differential response is a cornerstone of metabolic cancer therapy, allowing for selective targeting. Beyond simply starving tumor cells, DER exerts multiple beneficial effects. It is potently anti-angiogenic, inhibiting the formation of new blood vessels that tumors rely on for fuel delivery and waste removal, without the adverse effects of toxic anti-angiogenic drugs. It is pro-apoptotic, triggering programmed cell death in tumor cells through reduced glycolytic energy and activation of pro-apoptotic pathways, a crucial distinction from necrotic cell death that exacerbates inflammation. DER is also profoundly anti-inflammatory, suppressing key inflammatory pathways and reducing pro-inflammatory molecules, an unparalleled systemic effect compared to most oncology drugs. Finally, DER is anti-invasive, shown to reduce the metastatic capabilities of aggressive tumors like glioblastoma, a vital effect given invasion is a primary cause of mortality. Implementing DER often involves a restricted ketogenic diet, KDR, initiated by gradually reducing glucose while elevating ketone levels, with patients rigorously monitoring blood glucose and ketones to identify triggers. The target zone of metabolic management aims for low glucose and elevated ketones, levels shown to induce metabolic isolation and growth in tumors. Fasting can rapidly achieve this state in healthy patients. The strategy proceeds through phases: initiation, pre- and post-surgical support to reduce tumor vascularity and inflammation, and long-term maintenance, involving continued KDR or diet cycling, always with careful monitoring. This approach can be synergized with other interventions: anti-glycolytic drugs like 2-deoxyglucose, hyperbaric oxygen therapy, which stresses tumor mitochondria, glutamine-targeting drugs, especially for myeloid-derived metastatic cells, and therapies like chloroquine to inhibit tumor phagocytosis. By influencing the entire tumor microenvironment, DER and synergistic strategies restrain cancer progression at multiple levels. This holistic approach, termed mitochondrial enhancement therapy, MET, reframes treatment towards boosting normal cellular metabolic efficiency and resilience, moving away from a punitive restriction mindset.
While implementation faces challenges due to its non-conventional nature and the lack of integrated clinical support, the metabolic perspective empowers patients to actively manage their disease. It demands a shift in oncology to acknowledge and inform patients about these effective, non-toxic options, fostering a new era of personalized, metabolism-focused care. Moreover, the principles of metabolic health extend powerfully to cancer prevention. Avoiding chronic inflammation from smoking, obesity, environmental toxins, even subtle EMF exposure, embracing periodic therapeutic fasting or KDR, and prioritizing mitochondrial protection can significantly reduce cancer risk. By enhancing ketone metabolism and activating processes like autophagy, these strategies create an environment where precancerous cells are metabolically challenged and eliminated, delaying entropy and safeguarding organismal health.
Integrative Analysis and Implications. The accumulated evidence presents a profound challenge to the prevailing genetic paradigm of cancer, demanding a fundamental reorientation of research, clinical practice, and public health initiatives. The notion of cancer as an incomprehensibly complex genetic disease, characterized by myriad independent mutations, has proven to be a cul-de-sac, hindering significant progress against its most lethal forms. Instead, the overwhelming data converge on a singular, unifying principle. Cancer is a metabolic disease, fundamentally rooted in chronic mitochondrial respiratory insufficiency and a compensatory, pathological reliance on fermentation for energy. This metabolic framework offers a more coherent and parsimonious explanation for cancer's bewildering complexity. The genomic instability, aneuploidy, and diverse mutations, often cited as the root causes, are, in this view, downstream consequences. Impaired oxidative phosphorylation, crucial for maintaining genomic integrity and proper cell division, leads directly to the mutator phenotype. Oncogene activation and tumor suppressor inactivation are not initiators, but rather adaptive mechanisms, epigenetically upregulated or suppressed to sustain fermentation and cell viability in the face of respiratory damage. This explains why seemingly disparate carcinogens, chemicals, radiation, viruses, inflammation all converge on damaging mitochondria, and why even inherited genetic predispositions often link back to mitochondrial health. The implications for cancer management are revolutionary. If all cancer cells share a central metabolic defect, then cancer is not thousands of distinct genetic diseases, but a single metabolic disease with varied manifestations. This perspective provides a powerful universal therapeutic target: the tumor's inflexible dependence on glucose and glutamine fermentation. Current targeted therapies often fail to achieve lasting remission because they do not adequately inhibit these core fermentation pathways, allowing cancer cells to adapt and develop resistance. Immunotherapies, while promising in some contexts, may struggle against metastatic cells that are essentially derived from the body's own immune cells, macrophages, or their hybrids, making them less recognizable as foreign invaders and highlighting the need to target their unique metabolic vulnerabilities, such as phagocytic capacity. The metabolic approach, epitomized by dietary energy reduction and the restricted ketogenic diet, offers a non-toxic, broad-spectrum strategy. By exploiting the metabolic inflexibility of cancer cells while simultaneously empowering normal cells with alternative fuels, ketones, this approach selectively starves tumors, induces apoptosis, inhibits angiogenesis, reduces inflammation, and curtails invasiveness. It stands in stark contrast to the highly toxic, systemic damage inflicted by conventional therapies, which often exacerbate the metabolic dysfunction that fuels tumor growth.
Beyond treatment, the metabolic perspective profoundly reshapes cancer prevention. Rather than focusing solely on avoiding mutagens, it prioritizes protecting mitochondrial health. minimizing chronic inflammation, a ubiquitous trigger for mitochondrial damage, through lifestyle choices, diet, and environmental awareness becomes paramount. Periodic fasting and metabolically optimized diets are not merely adjuncts but foundational strategies to maintain cellular resilience, activate autophagy, the body's self-cleaning mechanism, and prevent the metabolic shift that precedes malignancy. The persistent stagnation in significantly reducing cancer mortality for advanced disease is a stark indictment of the genetic-centric dogma. A true paradigm shift is urgently needed, recognizing that uncontrolled cell growth is driven by insufficient respiration and its compensatory fermentation, not by aberrant proteins transmitting spurious signals. Embracing this metabolic framework promises a new era of effective, non-toxic, and cost-effective solutions for both managing and preventing cancer, ultimately empowering patients and transforming our collective approach to this formidable disease. The future of cancer therapy lies not in endlessly cataloging genetic mutations, but in understanding and exploiting the fundamental bioenergetic vulnerability that defines malignancy.
Actionable exercises. 1. Experience your metabolic flexibility. This book highlights that your healthy cells are remarkably adaptable, capable of switching fuel sources to thrive, unlike cancer cells, which are often stuck on glucose and glutamine. This exercise helps you personally experience your body's innate power. Under the guidance of your healthcare provider, consider a short period of dietary energy reduction, such as a 24 to 48-hour water-only fast or a very strict low-carbohydrate intake. During this time, observe how your body feels as it begins to shift from burning glucose to burning fat and producing ketones. You can measure your blood glucose and ketone levels with a home meter similar to what diabetics use to see this metabolic switch in action. This first-hand experience will not only build confidence in your body's resilience, but also deepen your understanding of how this metabolic difference can be leveraged against cancer. Chapter 2. Become your own metabolic detective. Understanding your current metabolic state is the first crucial step towards proactive health and potential cancer management. This exercise empowers you to gather personal data and identify your body's unique responses to food and lifestyle. Acquire a blood glucose and ketone meter, often available at pharmacies. For a week, record your blood glucose and ketone levels first thing in the morning and about 1 to 2 hours after different meals. Keep a simple food and activity journal alongside your readings. Pay attention to how various foods impact your glucose levels and whether certain patterns lead to higher or lower ketones. This simple practice will reveal your individual metabolic baseline and highlight dietary triggers, providing invaluable insight for making informed food choices that support mitochondrial health and starve potential cancer cells. 3. Fortify your mitochondria. Reduce inflammation. The book emphasizes that chronic inflammation and external stressors directly damage your mitochondria, which are the root cause of cancer's metabolic shift. This exercise focuses on practical steps to protect these vital cellular powerhouses and create an environment inhospitable to disease. Identify and actively reduce sources of inflammation in your daily life. This includes evaluating your diet for processed foods, excessive sugars, and unhealthy fats, replacing them with whole, nutrient-dense options. Beyond diet, explore stress reduction techniques like meditation, mindful breathing, or spending time in nature. Ensure you get adequate restorative sleep. Review your household products and personal care items for potential environmental toxins. By making conscious choices to lessen inflammatory burdens, you directly support robust mitochondrial function, enhance your body's natural defenses, and fundamentally reduce your cancer risk. 4. Craft your strategic eating plan. If cancer is a metabolic disease relying on specific fuels, then strategically limiting those fuels becomes a powerful intervention.
This exercise guides you towards designing an eating plan that leverages this principle, whether for prevention or management. Collaborate with a healthcare professional or a knowledgeable dietitian to develop a personalized, restricted ketogenic diet, KDR, or a carefully managed dietary energy reduction plan. Focus on prioritizing healthy fats, consuming moderate protein, and severely limiting fermentable carbohydrates, sugars, and starches. Learn to select foods that nourish your healthy cells while depriving cancer cells of their preferred energy sources. The goal is not just restriction, but strategic nourishment that aims to keep your blood glucose low and your ketones elevated within a therapeutic range, thereby placing sustained metabolic pressure on any metabolically inflexible cancer cells.
5. Optimize your total metabolic environment. Cancer isn't just about what you eat, it's about your entire cellular environment. This exercise encourages a holistic approach, integrating lifestyle factors beyond diet to create the most inhospitable environment for cancer cells. Beyond dietary strategies, consider other aspects of your daily life that can impact your metabolic health and reduce a pro-cancer environment. Explore regular moderate exercise to enhance mitochondrial efficiency and improve glucose metabolism. Prioritize consistent, high-quality sleep to support cellular repair and hormone balance. Discuss with your doctor if adjunctive therapies like hyperbaric oxygen therapy, HBO, which increases oxygen delivery to tissues, or supplements that support mitochondrial function or target glutamine metabolism might be appropriate for your specific situation. By addressing multiple facets of your metabolic environment, you create a powerful synergistic defense against cancer. That wraps up our exploration of today's book. If even one idea here gave you something to reflect on, give it a like so YouTube knows to share more with you. And if you'd like the next insight in your feed, hit subscribe. Remember, what you heard today is our independent interpretation, enriched with analysis and exercises. The full book has far more depth, so consider reading it to experience the author's work directly. Thanks for listening to DeftU Insights, where we turn great books into tools for growth.